Hello and welcome everyone to this podcast for cell biology. This podcast will be the first of several podcasts over the next several days that are going to be directed towards cell division. I wanted to begin this idea of cell division and the discussion of cell division with reminding us about the cell theory. Remember, the cell theory says that all living organisms are composed of one or more cells. The second tenet of the cell theory is that the cell is the basic unit of life. And the third one here is that cells arise from pre-existing cells. They don't arise spontaneously. There's always a cell and then it produces another cell. And really this is the focus of this podcast and the next several podcasts. How is it that we're going to go from one cell to two cells? We're going to talk about how this is a very organized, regulated sequence of events that does several things. It duplicates, and so duplicates the DNA and all the other stuff in the cell, and then segregates it to two cells. It does this by two special kinds of cell division. One would be mitosis, which is what we're going to focus on in this class, and the other is a specialized cell division called meiosis, which is more appropriate to talk about in genetics. But mitosis produces, as it does this, it will produce identical genetic cells here. Meiosis, when it makes new cells, it creates considerable genetic diversity. So I want to spend a little bit of time talking about the time it takes to go from one eukaryotic cell to two eukaryotic cells. And one of the fastest cell divisions would be our embryonic cells. They will take about 30 minutes to divide. They divide really fast. Whereas yeast cells, also a eukaryote, but there's a single cell, they take between 1.5 to 3 hours to divide. Epithelial cells like intestines, so the epithelium of the intestines, they divide fairly fast for eukaryotic cells. They're about 12 hours. Other cells, like a fibroblast cell, will take about 20 hours to develop. And then still we have cells like a neuron. Neurons don't divide, so we'll just say 0 hours, because they don't divide. So we have this wide range of cells and how fast they divide. And we want to talk about how a cell goes through this, and we'll touch on some of these timing events, but more importantly, how it goes about duplicating and segregating the material. So let's talk about the cell cycle. So the cell cycle can be divided up into two major phases. The first one is M phase, and the second one is a very long phase of the cell cycle, and this is interphase. Now, interphase itself can be divided up into three sections: G1 and G2 on either end of the M phase, and then S phase. All right, M phase, I'm going to indicate real quick here that it's about 1 hour. That's about how long it takes. Interphase, depending on the cell type, but it can be greater than 20 hours. All right, I'm going to erase interphase here because I want to focus on the individual parts of G1, S, and G2. G1 here stands for gap 1. Gap 1 G1 can last about 11 or so hours, depending on the cell type. During G1, the cell grows and it monitors the internal, the cellular environment, and external environments to see if it's receiving the signals necessary to go from G1 to S phase. In S phase, this is when the DNA is copied. This is when DNA replication occurs. Following DNA replication, G2 comes along. Following S phase, G2 is next. G2 stands for gap 2. Cell also grows during this time. And remember, it's important for the cell to grow because when you're going from one cell to two cells, you need to make sure that if you're going to make identical cells, they also need to be about the same size. So if the cell's not growing, you're going to produce, from one large cell, you're going to produce two small cells. It will also monitor the environment to make sure it's ready to go to M phase. but more specifically, it checks the DNA for mutations. I forgot to mention some times for S phase and G2. S phase is between 6 and 8 hours, about, and G2 is about 4 hours. So what's happening during M phase? M phase is the time when the DNA and the cytoplasm, including all the organelles and other molecules in the cytoplasm, are segregated into 2 new cells. And so at the end of M phase, as you're producing those 2 new cells here, after it's segregated into the DNA and the cytoplasm to the cells, we call that cytokinesis. And to draw it down here, cytokinesis occurs as these cells begin to pull apart from each other and fuse the membranes together to produce 2 of these identical cells. So this is the process of cytokinesis. Before we move on to the next slide, I want to stress how important this monitoring of the environment is for G1 and G2. This is how the cell will ultimately decide whether or not it should move forward in its cell cycle. Everything's happening in the cell, but ultimately the decision, so to speak, of the cell to move on is based upon the internal and the external cues it's getting. Okay, before we move on to the next board here, I want to talk a little bit more about these times. And these times are estimates, but for the most part, DNA getting copied in a typical human cell is going to take between 6 and 8 hours. There's not too much that happens to speed that up or to slow it down. And same thing with M phase. But where we do see some examples of shortening or lengthening the G1 or G2 is something I want to spend a little bit of time talking about. And to talk about it, I want to talk about embryonic development. And I think it's going to be better for me just to show a picture here as opposed to trying to draw it. And one thing you'll notice as you look at this picture here is you're going from a fertilized egg all the way to a 20-week-old fetus. And what you may notice is that from this fertilized egg to the 2-cell stage, to the 4-cell stage, to the 8, to the 16, and really even to the blastocyst stage, what you might notice is that the overall space that is being consumed by these different cellular stages is about the same. And this is a fairly accurate depiction. The size of the fertilized egg and the 16-cell stage or the blastocyst is about the same. So how this occurs is by having almost no G1 and no G2. You still have a G1 and G2, but it's very much reduced. So you're still going to have your M phase, and you're still going to have your S phase. And their timing isn't going to be too much different. But what does happen is that this G1 and G2 is almost brought down to 0. So very, very short. And because you have a smaller G1 and G2 phase, you don't allow that time for cell growth, as we see here. And so essentially all that's happening is you're copying the DNA and you're segregating it to 2 new cells. Copying, segregating, copy, segregating. So what you end up with then is a small embryo, say at the 16-cell stage, that's about the same size as the fertilized egg. This is important because during that early embryonic development, cell division has to occur rapidly. And the goal is to make a lot of cells so that by the time it implants at this stage here, at this blastocyst stage, that there's enough cells to carry on with that embryonic development. So it's important it goes fast, and the result of going fast is that you have many cells that are small. So how does the cell know, say during embryonic development or in a fibroblast cell versus a neuron, where to stop and when to continue a cell cycle? So that's going to bring us into our next topic, and that topic is the cell cycle control system. So let's talk about the cell cycle control system. The cell cycle control system is a highly regulated system of proteins that ensures that one cell cycle phase is complete before the next one begins. This cell cycle control system is broken down into 3 cell cycle checkpoints. So to talk about these cell cycle checkpoints, let's draw our cell cycle where we have G1, S phase, and G2. The first cell cycle checkpoint occurs here, and we call it the G1S checkpoint. Here, the checkpoint determines if the environment is favorable. So I mean external and internal environment. So if we have a cell here and it gets a signal from outside of the cell that says the system needs more cells, then that will trigger this to go from the G1 to the S, so it can begin the process of making 2 cells. However, if there are already plenty of cells, that signal isn't going to come to the cell to divide, and so we won't make more cells. So it assesses whether or not, based on cues it's getting from the outside and also cues that it's generating on the inside of a cell, if it's ready to move on to the S phase. If it's not, it will exit the cell cycle, so to speak, here and enter this G0 phase. At G0, it will just rest there until it gets some kind of cue to re-enter the cell cycle. But some cells will stay here indefinitely, like a neuron. Other cells will stay there for a specific set of time until they are cued to re-enter the cell cycle. Some of these signals that it could get could be molecules from a cell signaling pathway, could be nutrients. But if it receives that cue to move on, this cell here will now enter into S phase. In S phase, it will copy that DNA into 2 copies. It will enter into G2. In G2, the cell will get bigger, still with those 2 copies of the DNA. But before it goes into M phase, we have another checkpoint. And this checkpoint is called G2M checkpoint. Not very creative, but very descriptive. This is a very important checkpoint as well. And what's happening here is the cell determines the following. Is the DNA copied? Are there mutations? And if there are mutations fixed? There are times that the cell will encounter several mutations and it determines it can't fix them. So sometimes the result is one of 2. It may go through apoptosis and the cell will die, but if those errors can be fixed, and they are fixed, it moves past this checkpoint into M phase. We haven't talked about M phase yet, but we will talk about it later. But this is the mitotic spindle checkpoint. What happens here is, and when we talk about mitosis, it'll make a little bit more sense, but during mitosis, as the cell is beginning to pull those chromosomes to separate poles of the cell, each of those chromosomes have to be attached to a spindle fiber. So the spindle checkpoint determines if all chromosomes are attached to a mitotic spindle fiber, which is a specialized form of a microtubule. If they've all been attached, mitosis will continue and the cells will complete mitosis, separate through cytokinesis, and produce the 2 cells. But in order to accomplish these checkpoints, we need to talk about the proteins that are involved. Which proteins regulate these checkpoints? So let's talk about those next. So I'm going to redraw the cell cycle over here just so we can reference it if we need to throughout this discussion. And let's remind ourselves where these checkpoints were. So there's got to be a way, as I said on the last whiteboard, to tell the cell to move on to these different stages, these different phases. And it's largely going to be controlled by proteins. So we're going to need to have some kind of a molecular switch. And we've talked about molecular switches before. We've talked about GTPases and GTP binding proteins, as well as kinases and phosphatases. These switches that are going to regulate these checkpoints are kinases and phosphatases. A while back, when I talked about or introduced kinases and phosphatases, I indicated that kinases turn things on and phosphatases turn things off. Well, this isn't entirely true. A lot of systems will use kinases to turn things on and phosphatases to turn things off, but sometimes, as some of you may have learned in biochemistry, the reverse occurs. And in a moment, we're going to talk about how activating this system will require both kinases and phosphatases. But you'll have to listen a little longer before we get to that. But I did want to introduce this here because I've implied in the past that kinase is on, phosphatase is off. And I wanted to add to that concept that that's not always true. We do have instances where the reverse can be true. Now within a given cell, if I were to draw a cell here, it's going to be full of these enzymes, these kinases, that regulate the cell cycle. They're always very abundant in the cell. And if we were to draw that in a graph, this is what we would see. And I'm going to draw the kinases in brown on this graph. So they're always fairly high, but they're not always active. So over here I'm going to put kinase activity. And we know that kinase activity, and I'm going to draw that in red, has peaks. If kinases are always at a fairly high abundance, how is it that the activity goes through these peaks? They go through these peaks because kinases are not always active. They need to be activated by something. And they're activated by another protein called cyclin. And over here, let's put cyclin presence. And what we see with cyclin presence here, it will go up as the activity goes up. And then as the activity goes down, the cyclins will decrease. They begin to peak a little bit before the activity occurs, and then they drop once the activity stops. These cyclins are required to bind to the kinases to activate them. So these kinases that we talked about here, they are more specifically called cyclin dependent kinases. That is because our cyclin dependent kinases, in order to be active, have to bind to the cyclin. So as these peaks go up with activity, that's indicating that the cyclin and the cyclin dependent kinases are interacting with each other. So why is it that we have these peaks? Well, these peaks will help explain the fact that we have different kinds of cyclins and different kinds of cyclin dependent kinases. They will be specific for different parts of the cell cycle. So for instance, maybe we're looking at or drawing here a mitotic specific cyclin and CDK. So maybe what we're looking at here is a cyclin and cyclin dependent kinases, which we usually abbreviate as CDK. So this particular example, these cyclin and cyclin dependent kinases are only going to be active during mitosis. So this first peak here is mitosis. The second one is also mitosis. And the third one is, shockingly, also mitosis. And what's happening in between here as the activity goes down, this is interphase. Because during interphase, we don't need to move past the M phase, so the kinases are not going to be active. Or the mitotic kinases are not going to be active. They'll still be present, but they won't be active. So as I mentioned on the previous slide, there are several types of cyclins and CDKs, the cyclin-dependent kinases. And they're going to be specific for different stages of the cell cycle and be activated at precise checkpoints. So I want to draw here a slightly different figure. I'm going to draw G1 here, S, G2, M phase, and then repeating the cycle here. Now remember, the CDKs, cyclin-dependent kinases, always present. They just need to become activated. And so they're going to wait for specific cyclins to become present. And once they do, they can become activated and push the cell cycle past that checkpoint. So for instance, an S cyclin, the cyclin that's important for activating CDKs during the S phase, are going to increase right before the S phase. And they'll actually stay pretty high even through G2, but then they slowly will drop before M phase. So this is S cyclin. And though I'm not showing it, as it increases, it will bind to the CDKs and we'll start to see activity. And we'll come back to that in a moment, how we're going to activate and see that activity. But just to show the dynamic nature of the cell, we know that we're going to need specific cyclins to be present during G2 and during M phase. And so they also need to be turned on, but at the correct time. So down here we don't have very many M cyclins, but during G2 they're going to slowly start to be expressed and remain expressed throughout M phase and then drop off. So these are M cyclins, cyclins that are specific for M phase and the M phase checkpoint. So I should draw here that during S phase, right, so this S cyclin will activate the specific CDK and it'll remain active during S phase. And then during M phase, when that checkpoint is needed, the M cyclin will activate the CDK. So a few points I want to make here. So as we go from having no cyclin to having cyclin, we know that we have to have that gene activated. So it has to be transcribed, TXN, and it has to be translated. TLN are my abbreviations. So that gene has to be turned on. We also know over here, going from no M cyclins to having M cyclins, that we also have to have that gene for M cyclin to be active. So that gene is first transcribed and then it is translated. We then see this big drop of S cyclin and of M cyclin. And what happens here is that what happens here in both cases, but I'll just talk about it with S cyclin and CDK, is you'll have your CDK here and your S cyclin activating it. As it begins to drop off, what happens is that ubiquitin is added to this S cyclin. Ubiquitinating a protein like S cyclin will cause it to be degraded. It will go through proteolysis and destroyed. And so S cyclin drops off because it's being proteolyzed, and that then inactivates the CDK. So on this downswing here, we see proteolysis. Another thing on this diagram here is that we see S cyclin slowly moving up in concentration, but it doesn't immediately activate the CDK. Slow increase in cyclin, but then at some point the activity happens abruptly and all of it is activated. So what happens here is that you have our cyclin shown here, that's inactive, and in order for it to bind to the S cyclin, the cyclin itself has to be phosphorylated. So there is this kinase that's called Wee1. It's a kinase. And it will add a phosphate group to the cyclin. As it adds that phosphate group to it, it allows the S cyclin to bind. This is what we see here, this bound form is what we see here and during this time. And at this time, it's inactive. We call this an inhibitory phosphate group. It allows it to form, but doesn't allow it to become active. So now at this point here, before it becomes activated, all of the CDK is poised in this position here, ready to be activated. And we know that this occurs in a very abrupt manner, like turning on a switch. And so what happens is this phosphorylated CDK cyclin is dephosphorylated. And it's dephosphorylated using a protein called, or an enzyme called, CDC25. And this is a phosphatase. And that removes that phosphate group. And so now what we have is the bound CDK to the cyclin. And now it is active. And that's what we see during this phase of the cell cycle. The CDK and the cyclin bound together in an unphosphorylated form. And so this is what I talked about at the beginning of this, or near the beginning of this podcast, where I said sometimes phosphatases can be used to actually activate proteins. The last thing I'm going to talk about with the cell cycle control system are these CDK inhibitors. These CDK inhibitors allow for a quicker response time. They eliminate the need to transcribe a gene and then to translate that mRNA into a protein. And they do so by taking a CDK, like so, that's bound to a cyclin. And so, as written here, this should be active. And it would be active. But what can bind to this activated CDK cyclin is a protein. I'm just going to circle it, like so. But this protein covers the active cyclin CDK to inactivate it. So now it's no longer active. But the convenient thing about this is that when we want to activate this specific CDK cyclin, all we have to do, or all the cell has to do, is to dissociate this inhibitor that I've drawn here in purple, and now it falls off, and you end up with your inhibitor protein not bound to the CDK cyclin, which will now allow it to be active. This is a much quicker way to go about activating cyclin CDKs without having to transcribe the gene and translate the messenger RNA. Okay, that's all I have for this particular podcast. In the next podcast, we'll talk about G1, S, and G2 and the role mitotic factors, mitogens, have in the progression of the rest of the cell cycle. If you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.